We are live now.
Okay. So we got to start. Good afternoon in India, all the viewers, and on behalf of our team of international affairs and outreach IIT Indore, and on behalf of IIT Indore, so let me welcome all the viewers and especially today's speaker, Professor Devashish Naik from Biological Sciences Department, IISER Bhopal, and the happiness within us is he was a few months back, he was our own colleague, so it is a different pleasure of welcoming him. And of course, uh, the bad part is we missed him from IIT Indore, maybe for a better cause, we don't know, we'll see in future. So let's welcome Professor Devashish Naik and all the viewers of today to this public outreach event, Nobel Prizes 2021. And we are on the last day today. And so now I invite my colleague, Professor Prashant Kudgire, to chair the session. Yeah, Prashant, please.
Okay, thank you, Professor Sahu. Welcome, Dr. Devashish. It's a pleasure to have this particular session, the last lecture, public lecture in this series. We started this series last week. Inauguration was there on Friday. We had a lecture in physics, and then after the weekend, we had other lectures. And today it happened to be the concluding lecture for this particular series for Nobel Prize Awards in year 2021. Now, I don't need formal introduction to Devashish. Everyone knows Devashish. Devashish completed his bachelor's and master's in veterinary sciences. Then he moved to United States, completed his PhD in virology from University of Nebraska. Subsequently, he was there in one of the very premier research institutes of US, NIH, National Institute of Health. He did some wonderful research over there, mainly in virology and neurosciences and immunology interface. And then subsequently, he joined IIT Indore, our Department of Biosciences and Bioengineering. And he did some phenomenal work here on mainly animal viruses and virus-like particles, introduced several new courses here. And as Professor Sahu was telling that we miss you here, and this is a wonderful opportunity to recatch again. So today, Devashish will discuss about this year's Nobel Prize, and these are some very fundamental discoveries done where we are able to understand how we human beings or in general animals or cells are able to sense, whether it is sense of touch, whether it is sense of temperature or others. Many of this research involves understanding the receptors and how they function. And I'm sure Devashish will try to explain it in less technical or non-technical language. So I welcome you, Devashish. Floor is yours. Thank you very much. I just give one suggestion that we'll try to have all the questions at the end. So if you have any questions, you can type in the chat section. We will have all these questions at the end. Devashish, over to you. Thank you.
Yeah, I'm sharing my screen. Okay. I hope it is now visible.
Your display is visible. Yes.
Okay, so I load my PowerPoint.
It's full screen now. Go ahead.
Yeah. So yes, Dr. Professor Sahu and Professor Kodgire, thank you for your kind words and most importantly, a generous introduction to this. And it is always a pleasure to interact with you and all the students in the community which I belonged before and also belongs to. And with this, we will go through our journey to know about why this topic is so interesting and why the award is given in a particular fashion, that how we perceive the sense of touch and heat as well as pressure. So with this, I once again thank the organizers for this opportunity and let us talk about the topic. Okay. So I wish you all a very happy and prosperous New Year, and I hope we'll have a much better, exciting days ahead compared to last year's corona-induced lockdown. Okay, so the two prominent scientists which sharing this year's prize are David Julius and Arden Potaposian. So both are the kind of persons who put fundamental questions ahead of any other than that. So they are persons who are pretty much curiosity driven and wanted a very basic question, that why it happens and how it happens. What is the mechanism of that process, which is the process of sensory receptors present to identify a nature of thermal, such as heat, or a pressure, which is your piezo or similar kind of torch or pain. So before I go to that, I just want to give a brief overview of their career. David Julius, which is currently heading the department at UC San Francisco, has his PhD done in UC Berkeley. Subsequently, he did his postdoc in Columbia University of New York, and since last 22 years, he is at San Francisco. And while Arden Potaposian, he is a neuroscientist, and he's also PhD from California Caltech, postdoc at UCSF in the same department where David used to work. So they are connected in that sense that both are there in California. Okay, and both of them are also Howard Hughes investigators, which is a very prestigious fellowship in USA, which grants money based on your concept notes, and so that you can carry forward long-term research, 5 to 10 year research. So their seminal discovery gives us the very basic and fundamental view that how we sense touch, pain, sound, blood flow, and all the basic physical and interfaces we look at it are the seminal work of these two individuals. Okay, and what drives this discovery towards Nobel Prize is that not only they understand the fundamental biological process of communication, that how external stimuli such as heat is being translated to a neurotranslation in terms of electric pulse through a particular channel which is called ion channel, which we'll discuss later. And what are those applied parts, like how can you use that information to bring a honor to the humankind to go ahead with drug discovery or produce some of the treatment or therapeutic avenues which subsequently helps the humankind. So a basic science-driven, curiosity-driven research has a fundamental application for the humankind in terms of therapy as well as disease treatment. So that is the nature of their work, and hopefully I will try to convince you of that one. And their seminal discoveries opens the door for new scientific discovery. So basically, it is a feed-forward mechanism, or we call explosive research. That one thing is done, then several multitude of lateral branch of this phenomena comes. These are some of the papers. Just I'm quoting from their work that how frequently and how convincingly they could provide it. So let us go to that topic. And before I go to the topic, for the easiness of this talk, I have divided the talk into two parts. In the first part, I will talk about David Julius' discovery. Subsequently, we talk about Arden Potaposian's discovery. So David Julius' discovery is towards the how papers or peppermint identified the sensory receptors for temperature and pain. Okay, that is the layman language, the questions we are now focusing on. So we know that from our childhood, everybody at 3 years, 4 years old, that we are taught very nicely that we have 5 senses. Okay, or panch indriya hote hain hamara. So we are the 5 sensors, persons or individuals in the natures. Okay, and this is one of the supreme quality of being a human, that we have 5 senses. And these 5 senses are like touch, smell, or hearing, taste, as well as eyesight, which is a vision. Okay, all these senses are centrally located in the root of the information flow, as well as the root of the outflow of the information is at your brain, and different parts of the brain they are specialized, having certain variety of neurons, which are the fundamental cells for the brain functioning, are sense those kind of informations from the periphery or outside world. So we call it as a peripheral nervous system, which is your in your hand, your body, or your ear, in your tongue, and that goes to the brain in a centralized way. We call central nervous system. The peripheral sensory organs or the somatic sensory organs are conglomerated in the brain, and where the information inflow and outflow is happening very nicely. And in theory that, as you know, that we biology people try to do something called knockout. Okay, so that means we throw out, or you break that connection of 5 senses, and the person becomes a nonsense person. Okay, so commonly most of you have heard the word that nonsense. Okay, but actually it is a derogatory word, and there is no such person called nonsense person, because in fact we cannot evolutionarily we are not able to knock out all the senses, because if it is done, the baby will not be born to the earth. So the baby is died in the womb if we knock out 5 senses at a time. So don't think that the nonsense exists in the real world in theoretical manner. Okay. So before the discovery of this seminal work by Arden Porte-Passian as well as the David Julius, there was a series of fundamental discoveries which led the time for these two people or many other people to ask other questions. For example, in 17th century, a philosopher Rna Descartes, who theorized or philosophized that if you touch a fire, okay, your body will go for a series of events, and that information will go to the brain. So we are talking about 17th century, when there was no basis of any molecular approach or any experimental control or any kind of intervention to the experiments. So the theory remains as theory, and it was. But the major contribution happened in 1906, where two people, which we already know, that Camille Golgi, which is also an Italian scientist, as well as the Raymond Carswell. Okay. So these two people gave a very similar discovery, where Golgi proposed that the sense of this information, which we call nervous information, are being a organized network inside the body. Okay, and he tried to find those things in the body by staining various cells using simple silver nitrate, which we still use it today to identify the neurons. Okay, and also here discovered another organ we call Golgi body or Golgi body. So this is his part. And similarly, Raymond Carswell, which is also a fundamental scientist, he tried to tell that these informations are being, like a brick and mortar, the information which has been circulated inside the body are because of the neurons. So neurons are the fundamental cells who carry this information inside the body. And this very small and very specific discovery led to the Nobel Prize in 1906, which is here I'm showing you. Okay. Similarly, in a couple of decades later, the Charles, Edgar Adrian, these are the two people who fundamentally look at the function of these individual neurons, which Raymond Carswell discovered. So how the neurons whether they have a motor end, or whether they have a body and a somatic, then that synapse. So these are the individual part of the neuron, and we know that neuron to neuron connection will transmit the electric pulse like that of a electric wire current type, and that individual activity of a neuron, which is collectively give to the nervous system, is the fundamental work of this Charles as well as the Edgar Adrian, and for that reason also they received Nobel Prize in 1932. And the last most important discovery in this series was by in 1944, where we have Erlanger Gasser, who discovered the highly differentiated function of the single nerve cells. So what they mean that, so here everything was going as a neuron, but these people try to identify the neurons which are present in your brain, which can sense a smell, or neurons which can sense something else, such as a heat or your pressure or your cold or temperature are different. So this much of information, he could tell that all the neurons are not equal. Rather, they have a very specific function, and each function is carried by a specific fibers in the nerve, and this how they got into this discovery and this thing. And so far, this has been the course of the discovery regarding the sense and touch. In between also, there is a lot many things happen, and these are the natural elements such as opium, okay, which we know infamously that opium contain a alkaloid we call morphine, okay, and you make a further derivative, we can make heroin, and which is 1 crore rupees per kilogram at this moment. I don't do that. Opium is a very specific substance which is highly addictive, okay, which is a very solemn work by Snyder, who discovered the opium receptor. Why it is important? Because opium receptor is a receptor which you block it by agonists such as morphine, you can block your pain. That means if a person is suffering from pain, by use of morphine or derivatives of morphine, he can be relieved of pain. So you can imagine that when there is a cut in your hand or your body is burned or there is an accident or surgery, how can you use that particular information other than what we talked before, that this specific information can be used medicinally as well as. And similarly, we have also cannabis and marijuana, and I don't want to go into that detail, but the fundamental thing that the natural substances, they do something in the human body, and you look at the mechanism, you find a wonderful thing. Okay, and this natural substance could be your chili, okay, which give you hot sensation, spicy, or it could be mint, which you also find in a peppermint or pudina or mint leaf, or simply give a hug to your daughter or son. So these kind of sense which we are getting is a derivative of a ultra modern chemical reaction going on inside the body, which gives a very nice feeling, pleasant feeling, readiness feeling, or hot feeling, whatever you feel about is these things. These are all natural things. We are not talking anything different. So there are a series of these events which happens around us, okay, inside us, and we get the sense, but yet we don't know how exactly that is happening, okay, quantitatively and qualitatively. Okay, we know it is happening, but what is that basis of that? Okay, so the environment which you are surrounded and the reception of this information through various senses, the mechanism is the fundamental work of these two Nobel scientists which you are going to discover. Okay. When we talk about these 5 senses, and we clarified that the 5 senses are like olfaction, which is smell, okay, which is a chemical way we are sensing it. Taste is also another chemical. When you talk about vision, it is optically, okay, which is physics, which is a photon. Light is now interacting, making certain rearrangement inside the electrode system in the nerve, where the synaptic system is getting the electric pulse so that we can see something. Hearing is another example where mechanical pressure or piezo sensors in your eardrum is translating to a certain information to your brain so that you can hear me and vice versa. Similarly, other way we have is touch, okay, which is either mechanical or it could be thermal. Okay, so you can close your eye, and if some object is given to you, you can easily tell that it is either room temperature or cold or hot. Okay, we do not need to see it, because that the sensor or molecular thermometer inside your body is the work of this scientist. Okay, similarly, touch is also similar way that how you can touch a thorn or a pin, or you can soft substance we can know from by touching, and our fingers are so sensitive, or so-called, you can create a sensitiveness inside your body that you can even differentiate 1/10 of a hair thickness if you are really practicing that sense things. So those mechanical as a thermal as well as the photon chemical, they all belong to outside the world where we sense and we live, and this is essential for any individuals to live in the earth, whether we are a human being or a plant or an arthropod or a monkey. So this must be there. Okay. So now let's focus on one particular aspect of this sensor system, which is called taste. Okay. So we know that taste varies person to person, but there are certain individuals or certain items which are universal. For example, if you take a chili, which I'm showing here, you will definitely feel hotness. Okay. Similarly, if you take a mint, you'll feel soothing effect in your mouth. Okay. So when you talk about this chili, so there is a specific chemical or molecule, we call it, which is natural molecule, we call capsaicin. Okay. So capsaicin is present in chili and some minor amount in your black pepper, rather black pepper cotton piperine, which is another substance. So both of these products, which I'm showing you the molecular formula here. Okay, just so here is your piperine, and here is your capsaicin. Okay, fundamentally they are natural products. Your red chili contain capsaicin, which is black pepper contain piperine. Okay, so black pepper contain 8% of the piperine, while red chili contain only 0.01% of the capsaicin. So you can imagine that capsaicin is so hot compared to piperine, though they taste hot, and one capsaicin molecule equal to 100 type, or you can say that hotness of the capsaicin is 100 times that of a black pepper piperine. So that is the message we want to tell, that how these two products discovered the Nobel Prize worth of organism. So we know that in your brain, there are different parts of the brain which is specializing in a very specific function. So you need not to be a neuroscientist, but believe me, there is a part of the brain we call dorsal roots. Okay, and in the dorsal root, which is the back side of the brain, so there is a set of neuron. When you have a set of neuron, we call it ganglion. Okay, so ganglion is a collection of neurons together. This dorsal root of ganglion is now connected to your taste bud inside your mouth or tongue, where you can sense your capsaicin or black pepper piperine. Okay, so the point is that we know that in your mouth, when you put the piperine, you feel hotness. But how that happens? Okay, why other things do not cause that thing? Why you take paracetamol or sugar, it does not make it? Okay, what is the molecular basis of that? Okay, so this discovery is primarily done by David Julius. Okay, and that we are going to talk to. Okay, so what happened when there is a signal transmitted inside the nerve? Basically, that signal, we call it as the electric pulse. Electric pulse, if you remember, the electricity flows from one end to other end when there is a difference in the gradient of a voltage. Okay, so the voltage is here in millivolt. Okay, and the voltage we call in physics terms also potential differences, or PD. Okay, and the potential difference is achieved because a set of ions, particularly here positive ions, which is your sodium ion as well as calcium ion, which is a bivalent ion and monovalent ion, they change their places from one place to other. Okay, so when they are in equilibrium, but they go from one place to other, then there is a difference in the potential difference, or voltage change is happening, and that voltage change is producing a flow of current, and that flow of current is subsequently transmitted to a set of neurons to the brain, and where you can again reconstruct it as a hotness or coldness, or a light or something like that. The basis of this information is the ion channels, which operate in a particular stimuli, and that ion channel produces current, and current is now passed to your brain. And if you correlate these things to modern discovery, which we know Elon Musk is putting heavily amount of money in a particular company called neural network. Okay, and he is trying to connect your brain to external devices, where the external devices can operate by signal of the brain, and you need to talk, you need to operate your hand, all you need to do, pass the information to the external device, and that will, suppose you want to type something, you just think of it typing, and computer will type itself. Okay, so this is because of this very specific stimuli, and which is unique stimuli and extremely low in quantity, that operates inside your brain, inside the body, and that is where we sense things outside. So the question is, what David Julius asked is that if you put capsaicin, which is your paprika or red chili or hot chili, how does that thing is being perceived by your tongue to the brain? What is the molecular basis of these things? Okay, and that is where he hypothesized that. Okay, so we know that the curiosity-driven science is based on hypothesis, and it is utmost important that we should believe something. And students, I always try to tell you that hypothesis is a unidirectional statement. Okay, so that means I hypothesize that there will rain in Indore today. Okay, there is no question of may rain, may not rain. I hypothesize there will rain. You can prove me wrong or right, depending on the time. But This is my hypothesis. Okay, you hypothesize there will be no rain. We are equally competent to prove our cause, but you cannot say that I hypothesize that there may be rain or may not be rain. When you say may or may not, these are not a scientific term. These are layman terms. So whenever you write a statement in your thesis, you tell that I hypothesize that. Okay, if capsaicin receptor exists in the mouth, which David Julius hypothesized, if it is there, then we can clone it. We can make a copy, and from a nerve cell, we can bring that information, put in a liver cell, and reproduce the same thing in the liver cell. So fundamentally, what he hypothesized that if the neurons have this receptor, which only neurons have it, your liver cell doesn't have it, your skin cell doesn't have it. If somehow can identify that receptor and put in a fibroblast cell such as your skin cell, your skin will also behave or can send the test of papain. Okay, that is his hypothesis. Okay, what he tried to do that he tried to use a very specific and proven technology to identify the gene receptor, like cloning every gene which is present in the neuron. Okay, so he did not give any chance. He tried to find the cause. So there are about 20,000 genes in the neuron. Okay, and he cloned all of these things. Okay, and each gene he cloned, and each gene he tried to put in a fibroblast cell and try to ask the questions whether the fibroblast cell can now sense this paprika or the capsaicin. So this is the set of the experiments what he did that. Okay, but we know that when somebody hypothesizes, there is an inherent risk. Okay, because he is going to a very specific direction, unidirectional direction. So he hypothesized that there is a receptor. What if there will be a multiple receptor? Then the experiment will not work. Okay, because you are cloning only one. Sometimes you know that coronavirus goes through a receptor called AC2 receptor. If it is present, corona goes in. If it is not present, corona doesn't go in. This is very, very fixed. But think about HIV. Okay, it needs CD4 receptor, CR5 co-receptor. So there is a multitude of receptor. Sometimes a receptor is also made by a couple of genes. Okay, so what he did that he cloned one gene, but he did not know that whether it will work or not. But these are the risks. Okay. So the risk here is that the cell should have one receptor, single gene should clone only that receptor, and there should be no co-receptor. If these three things are there, assumption is right, then the hypothesis what he proposed will work. Otherwise, if any of things is not right, things will not work. Okay, so that is the risk of the experiment, and we know that he did the things in 1970s, and where things are not ordered, and he collected all the 20,000 gene information and tried to clone in other cells. Okay. And in science, sometimes we have to be lucky, and sometimes we go with that with risk, and that's how the nature of science is. Everything is not predictable and not that. Okay, so to do this experiment, what he did that once he cloned that particular gene in a fibroblast cell, so and he could made a very tiny micron size electrode. Okay, so we know that the electrode has a tendency to measure the potential difference. So that means if an electrode is outside the cell versus one electrode inside the cell, then there will be change in the velocity which or the voltage which can be shown or you can be measured. So if a receptor is there here, okay, and you put capsaicin, then there is a voltage change. Okay, so for each 20,000 gene, he tried to do one after other and after like that. And in his postdoc, which is Makoto Tomiyaga, he actually did the work in the 6th year. He could complete the entire screening, and he found that there is such receptor in this system, which is present in the cell membrane. Okay, here, and that gives the signals, and the voltage is changing. Now they could identify the single molecular mechanism or the presence of a specific receptor which respond to the capricorn, which is a capsaicin, which is your hot chili. Okay, and they named it at TRPV1. Okay, and subsequently there is more work has been done. So what else they found that the same receptor also respond to a temperature when the temperature is above 43 degrees centigrade. So that means you feel that chili is hot, okay, and always say that hot, but actually this hotness is also a scientific way that the receptor is sensing that if you put 43 degree element to your mouth, the kind of sensation which you feel, okay, will be similar to that of a hot chili, okay, at room temperature. So the same receptor which behaves differently, behave similarly either to heat or to the capsaicin, and they allow the sodium and calcium to go inside cell from extracellular environment to inside the cell, so that there will be net potential difference, okay, which can be captured by this electrode. And this change in electrode voltage will tell that how strong your chili is. Okay. So to summarize this particular work, what we say that using a in vitro system where they could clone the gene of interest which they identified from the neuron, and they could go one after other and screening 20,000 genes, they could find this particular receptor called TRPV1. Okay. Subsequently, there are many other people who verified this information around the world. Once somebody discovers one thing, others also questions that one, and that is where the science moves incrementally. So particularly Europe, Japan, as well as in Canada, people also try to clone that receptor because it's a very small receptor, okay, about 98 kilodalton or so, and it has 6 transmembrane protein, okay, and that means each protein has a beta helix. If somebody coming from protein background, so we know that the protein has a kind of amino acid which will love to be in the membrane, which is lipid. We call it lipophilic, and some of the amino acids are loved to the water. We call hydrophilic. So hydrophilic is lipophobic or vice versa. That means if a molecule is hydrophobic, they're trying to stay in the lipid membrane, which is in your body or plasma membrane. So these receptors are engaged in the lipid membrane in your plasma membrane cell in neuron, and they have this external outside layers which can sense the heat or capsaicin, and subsequently by cryo-electron microscope structures, okay, so they identify this 95 kilodalton plasma membrane protein to be the receptor for heat as well as the capsaicin. Okay, so I will give another limelight to this technology that to know the crystal structure. I think we know that the latest technology called cryo-electron microscopy. That means cryo means which you are putting at a temperature of ultra low. For example, liquid nitrogen, which is at minus 196 degrees centigrade. It is higher than the Kelvin temperature, which is minus 270 degrees centigrade. If you put any substance at Kelvin temperature, which is 0 Kelvin, it doesn't have any biological or physical property to remain frozen. But certain substances which are also temperature sensitive could be frozen at lower temperature. For example, such receptor can be frozen at minus 196 temperature, and that also 2017 Nobel Prize also awarded based on the discovery of this cryo-imaging electron microscopy, where you crystallize the protein or you freeze the protein or any substances, and provide X-ray to that substances, and there will be a lot of images produced because the angle at the X-ray will be diffracting. It will produce a subsequent image in your screen, and when you collect thousands of such images by specific software algorithm, we can collect individual information and produce the three-dimensional structure by computational biological approach, and we can construct the structure of the protein based on this cryo-electron microscopy, which is roughly 2 to 3 Armstrong in resolution at this moment. And in India, we know that NCBS had this facility, and now IIT Delhi has this facility. Subsequently, a couple more institutes, particularly IIT Kharagpur, is going to have this kind of facility where you can instantly go with a protein at a very low concentration, and or a substance at low concentration, and freeze it, and record your information and produce the three-dimensional crystal structure of it with a reasonably high accuracy and resolution. Okay, once this information is known, then many other scientists try to look at their nature of opening and closing of this receptor. So here, when there is, as you told, that when it is open, there is calcium as well as the sodium ions are going in from the outside to the inside. Okay, there is a change in voltage. Okay, so this opening and closing is happening because of these two arms which are present in this plasma membrane, which is either change the heat or presence of capsaicin or change the voltage. Because if it is operating for the voltage, even voltage change should open it, or if there is capsaicin or delta T, which is your heat, these membranes will operate. And there are many other things which I don't want to go, but subsequently these are verified and by other scientists, okay, which is another solid publication that yes, what David Julius discovered is actually true, and they could clone these things and verified. Not only that, the C617 is the name of amino acid, which is cysteine. Okay. When they change to alanine, okay, then the temperature sensitivity is lost. Similarly, you can see that each individual amino acids, when they are changed in this particular receptor, there is a very degree of this sense of delta T or voltage or capsule. So this is about hotness. So it is not that all the things operates in the hot area. There are certain elements also which presents in the cold area, such as a polar bear or a penguin or a ice or a mint or a oil of wintergreen, or we call eucalyptol. Okay, so menthol. Okay, so these are the substances which give you a very soothing feeling. Okay, so if you remember that, if you are taking pudina hara in a cold ice water, how smooth it is after having a hot chili sauce. Okay, so these are the similar natural substance which, like capsaicin, produce the exact opposite effect in the same individuals or same neurons. And what is there anything different, or it's just opposite of that? So to look for that, again, the David Julius group. Okay, so they proposed, actually, is there any menthol receptor? Does it respond to the cold? Is it related to TRPV1, which we just discussed, or is there anything else? Okay. So in search of that, they could find another molecule. They call it TRPM8, okay, or TRPM8, okay, which is present very close to that of the TRPV1, okay, but it is not the TRPV1. When they look at the amino acid structure, they look at the crystal structure, they are different, but yet they are present in the same location of your plasma membrane. So that means, so it is like if your same hand can, same way you can choose ice versus a hot product is there in your plasma membrane. Only thing that they are a couple of nanometer apart, okay, which is very, very sensitive. So what to prove this, cold sensor, what they did that they made a knockout mice. Okay, so here is the knockout mice, okay, which we have a TRPM8 mice. So this side the temperature is 20 degree, and this side the temperature is 30 degree. So that means here it is cold, here it is relatively hot. So whenever the mice put the head, you can see that it is staying away because it is sensitive. Okay, it has all this receptor. Again, it is attempting to do that. Okay. If you put the n number of time, okay, all the time this mouse is stay away from the cold environment. Okay, what if we knock out that receptor? Okay, you can see the results in that same mice. Okay, it does not distinguish. Okay, it goes there and come here and just so basically the sense of temperature is gone once you knock out this receptor from this transgenic mice. Okay, so see, this is a very fundamental work, and unequivocally, David Julius now conclude that TRPM8 is the receptor for the cold, while TRPV1 is the receptor for your chilly or hotness. Okay. So like that, when he discovered this field, there are many people who there are certain groups in the lab or certain elements around the world. They always try to be top notch, and we show that each time that something comes, there another comes. Couple of back-to-back Science Nature Cell papers who discover that yes, there is TRPA1, TRPM8, then TRPM3, TRPV4, all variety of receptor, a set of receptor was found. Okay, and now we can tell that the set of receptor can sense from 0 degree till 60 degree. Okay, so here is the temperature scale, and here is also this some of the, for example, TRPA1 can similarly sense garlic or cinnamon, okay, as a cold receptor. Similarly, chili, as I talked to you, it is hot, okay, and menthol, which is here it is cold region. That's why if you have a soothing feeling. Similarly, we have camphor, okay, and oregano. So various natural substances which are sensed by these things are also sensing the temperature in the similar manner, and we have all this information collected by the series of activity from his lab, his postdocs, so they become independent, they started the groups, and last 15 years of the discovery of TRPV1, all the new receptors came up and the whole era of findings came. Okay, so with this, the first part of talk is that in summary, using capsaicin, which is a red chili, Julius group could find a very specific receptor called TRPV1 that can operate above 40 degrees centigrade, which is a heat sensor, plus it can also sense the capsaicin. Similarly, they also find another receptor called TRIM8, which can also sense your temperature at 20 degree and so. Okay. Now talk to a different side. Now these are the chemical sensors. Okay. What if we have other sensors called touch sensor? Okay. Okay, which is something like pressure sensor, like put a leg inside the pin and how you feel it. Okay, so you feel the pain. Okay, and actually pain is very good. Okay, if there is no pain, we call it there is no gain. But in biology, we call it if there is no pain, there will be no life. Okay, so pain is an essential survival mechanism. Okay, so there are two types of pain, and we know it. One is acute pain. Sometimes you cross your finger or got it, or there's a chronic pain when you have a long-term pain which is there for a long time. Okay, so whether it is acute or chronic, there must be some mechanism which body sense it, and that is the work of the ardent protopression. Okay. So unlike David Julius, which is go for screening each and every gene, this guy was relatively young and he could do in opposite way. What he did that he knocked out individual gene. Okay, so what is that? He put a technology, we call it RNA interference. Okay, RNA interference is technology which is similarly to that Pfizer Moderna vaccine, that we are injecting a RNA for a purpose to make a protein. Okay, so that protein is going to produce an antigen, and subsequently that is a vaccine. But the bottom line is there is a piece of nucleic acid which is RNA, and similarly you can use the RNA to stop a protein production. Okay, we call a small specific RNA which is around 18 to 21 nucleotide long. When you inject to a particular cell, this cell will not express that particular protein. So that guy was doing the gain of function, this guy was doing the loss of function. Okay, so these are the two different approaches, but again they're aiming for the same thing. Okay, so this guy again hypothesized that there may be some sensors which operates on the pressure. Suppose you hug somebody, you feel some pressure. Okay, or there is a pain by thorn, you feel a pressure. What is that pressure? Okay, and that name he gave in a Greek word called piezo. Okay, so piezo means pressure. So the piezo 1 and piezo 2 are the two important discoveries made by his group and lab. Subsequently, it opened up the new things. Okay, what, as I mentioned, that the whether it is any such piezo pressure, TRPV1, hot chili, the basis of is that there is a signal which is in terms of voltage is going. Okay, whatever is the pressure is sensed or not, the ultimate reflection is a change in the voltage. Okay, so this voltage again operates in the plasma membrane, and he could curate that if this is a cell which is yellow is your cell plasma membrane, and he also hypothesized that this receptor is also present in the plasma membrane. Okay, if this is plasma membrane, then when you provide the pressure, okay, then this pressure will make a signal. Okay, so before that, there are a couple of more discoveries. I don't want to go to detail, but G protein coupled receptors also present, which is also in dozens in quantity, as well as they also sense, but none of the G protein coupled receptor sense pressure or these things. So this is a fundamentally new receptor. Okay, so how this information goes, as I told you earlier, that opening channels, there is flow of ions, then polarization, depolarization, voltage change, and that is electric pulse in the neuron, and you can sense it. Okay. So he did RNA screening. So what it is done is that these are like small well plates. Okay, in 96 well plate, you imagine that which biologists are working in a well plate. You have a particular cell, and that cell you add only one RNAi. So there are 25,000 RNAi library. It's also available in isogopal if you want to work. Okay, so you put one RNAi to stop one gene. So it is a loss of function study. So you knock down one thing or you stop one expression and look how the phenotype is behaving. So like that, he screened all like 25,000 category of the cells, and he found that how he was doing. So he has a very sensitive poking, like suppose I poke you and you can feel the pressure. Similarly, he was poking the cells with a small tweezer kind of thing, in a very pin kind of thing. Okay, it will poke it. Okay, when there is poke, if there is a pressure, if there is a sensor, then the voltage change will happening. And he was measuring that voltage change, which is in 100 millisecond, and this is the millivolt. If the millivolt is beyond 10 millivolt, if the delta MB is more than 10 or delta 10 MB, it will tell the difference. Otherwise, it will be noise. Okay, so like that, you can see there is a higher change in the millivolt, but here it is not. So basically, so change in the voltage by poking the cells, and he could identify which gene is responsible for providing that information. So as you told us that he was doing the loss of function study, each cell will have a specific deficiency, and that deficiency is losing the phenotype or losing the function or not. He was trying to find it. Okay, so by his quest, he could find a very complex protein. Okay, we call it piezo-1. Okay, it has 24 structure which is come across to the membrane. So this protein is resided in the plasma membrane very nicely and steadily, and this is the three-dimensional structure. So what happened that when you apply the force, okay, here, because we know that the force is mechanical here, and it forcibly open the latch, and there is opening of this gate. As a result, more ion, which is sodium and calcium, are going inside, and there is a change in voltage. So this is a pressure sensor. It's not a chemical sensor. If you apply force, then it will open, and if it is open, there is a voltage change. Okay, and this is his fundamental discovery. But till now, everyone is thinking that every change which is happening in the cell is because of some chemicals, which is sodium, ion, or other capsaicin or something else, but not physical force. Okay, so this is his discovery. Okay, and in his discovery, we know that he could find two important pressure sensors in the body, and they are here, piezo-1 and piezo-2. They are again have different functions. I am not going to talk to detail, because if you want to understand that when evolution is making you healthy, it has a backup plan. So if you lose a piezo-1 sensor, piezo-2 sensor, you are becoming a diff. Okay, if you lose both of them, but if you are losing either one, still partially you can hear it. Okay, so remember that nature has created, because your pressure sensor is very extremely important. That's why you can hear things. If you can hear things, so that's why the nature has created two versions of these piezo sensors, which is piezo-1, piezo-2, which is a discovery by the ardent Potopotian group. So now we know that we can sense coffee, or we can sense a pressure, and do these things. Okay. Then how does it become a Nobel Prize? What's the thing? As I told you, that very simple piezo-1, piezo-2 can make a person deaf. Okay, and it's a fundamental different world of a person. Similarly, vision loss can make a person blind, and that is way different than what we know about life in the earth. Okay, so this small discovery, yet important discovery, has a big impact. Okay, so and based of this, things, people discovered so many types of pathways which operates, because from ear to brain also there is a channel of information flows, or from your hand to your brain there is a channel of information flows. Okay, the pressure which you are present in the ear will not be present in the same as in your gallbladder. Okay, so gallbladder, when you feel susu, because you feel there is a pressure. Okay, or you are going to sandas or paikna, you feel that something is there inside your body. So what is that pressure? Okay, because that is internal organ pressure, okay, which is different than outside organ. So this similar discovery, when you tell that is a pressure sensor which is present in your hand, or could be present in your liver, could be present in your heart, lung, all the side ripple effect created a series of discovery of receptors and mechanisms, and that has relevant to a skin disorder or CNS disorder or respiratory, cardiovascular, kidney, pain, diabetes, you name it. Okay, so the entire medical field now being excited to have new things to know, and not only new things to know, to intervene the new things. Suppose, as I told you, that if you are feeling pain, somebody can prescribe you morphine or heroin or similar substances. Okay, and that's how the medical science developed. Similarly, you can now have a pain in your stomach. What receptor blocker you name it, now there is a group of people who are now discovering that drug. Okay, or if there is a cardiovascular pressure change, what should be done? Okay, very specific and way, logical way. Now we can produce our effort to go into it and discover the things. So that is how this slide is not. But here, believe me, this I'm telling you that how based on their discovery, many molecules are now phase 1, phase 2, phase 3 clinical trials. The series of molecules, more than 120 molecules are there. So coming years, you will see that very specific receptor blocker or agonist or antagonist are coming to the market based on the discovery of these two seminal Nobel laureates, which opened the field for more receptor, more mechanism, as well as the more options to cure human beings problem. Okay, that is how this discovery is important. Not only that, people who are beyond human being, which all of us also look at the environment or the situations where other entities which are vertebrates like a fish or dog or frog, all of them have a similar kind of mechanism. Only thing that sometimes your TRPV1 operates at 43 degree, but somebody it operates as a 40 degree or somebody for 30 degree because there is change in the small amino acids. That's why the minor changes because we know that there are 883 genes or so 883 amino acids are present in a particular TRPV1, and if you change a couple of amino acids, the 43 degree could be 48 degree, or could be 44 degree, or could be 35 degree. Okay, so those changes are actually accumulated in the nature, and the nature we can see starting from your amphibians to snake or lizard or monkey. The nature has provided such receptor in different contexts as well as different forms. But fundamentally, it is an evolutionarily conserved set of receptors present all around us. So what I conclude to the current topic is that, so this kind of discovery is always curiosity driven. And a great example is that a goal-oriented driven when we are looking for a paper, okay, in your third year of a PhD, versus you are wandering and trying to find a new papers or maybe 10 papers, okay, we don't know. So this is a fundamental way the science move in the field. We always value the curiosity driven science, or always the fundamental way to find a new discovery. David Julius nor Arden Puttaberton thought that their discovery could someday find a cure for the kidney or cardiovascular disease, but it is exactly that phase 3 clinical trial is going on. Okay, so with that, I invite you to IIT Bhopal, and we do have a cafe coffee day in the IIT Bhopal. Okay, so next time when you come here, you debate that whether iced coffee, which is a M8 receptor driven sensor, or hot coffee, which is TRPV1, we can discuss, because a lot of things can happen over the coffee. Okay, and you can feel a sense of belongingness here because I'm there. Okay, and please join me, and if you have any questions, please ask. Thank you.
Okay, now we can have some questions if there are any. Ashish, you can escape your slides.
I'm trying to do that actually. Okay, yeah.
I really enjoyed this talk.
Thank you.
It was, although I'm a biologist, so I could follow most of it. So maybe non-biologists, if they are there, they may have more questions about this work. But as you likely said, that these were some very fundamental contributions done. The people were trying to understand that how our senses, which is the first part of the nervous system that works, and this has tremendous applications. We know that all those people who undergo surgery, there are pain related to pregnancy, there are pain related to delivery, there are pain related to even the toothache. So all these are there where these molecules are going up and down, and therefore this is what is happening. So yes, there are lots of applications. So I invite our students to please ask questions if there are any. You can write in the chat section, or you can type on YouTube chat section, because that is also there. This seminar is live on YouTube, on IIT website.
Prashant, in fact, why biologists and even physicists could understand these fundamental mechanisms. So thanks to Devasys. So bringing this, difficult domain of biology to fundamental understanding. Thank you, Devashish.
Yeah, thank you. It's a pleasure, as usual, to interact with you.
One comment which I would like to tell our students, masters and PhD students who are here, that this will not happen overnight. These are few decades of hard work by multiple different research groups. One have to have lots of patience to do something like this. So there is no instant coffee in science.
So there is no question. We can end the session. And thank you. Somebody wrote, and did not do animal sense time just like other things. Or you are asking another question. Yes, the circadian clock. I think last year there was a Nobel Prize on that. Okay, that how you sense time. Is that your question? Is?
Yes, sir.
Yeah. So the circadian clock is a inbuilt, sensor system in the brain. And someday I want to elaborate it, but yes, they have also the sensory system to know the timing of a year or a season or something like that, so that they can prepare for the next. And animals could sense it. Actually, whole world is sensing it. Okay, even some of the animals, for example, insects. If somebody is coming from a rural background, you can see that when there is expecting to the rain, there is some kind of ant flies. They will come out from their ground. They will fly it. Okay, so they could sense the barometers, because we know that in physics that when there is a pressure change, your barometer reading is coming down, and if there is a change, likely there will be rain. Okay, even our system is not so sensitive. Rather, animals are much sensitive to this system. For example, elephant can sense something. Human is walking from 10-kilometer distance, so we cannot. Okay, so they are very sensitive because their survival instinct is heavily dependent on the nature. Because their brain is relatively less, that's why they have to sense everything accurately as much as possible. So sensing the time as well as other things is extremely accurate in animals also, like us.
Thank you, sir.
Yeah, thank you.
Any question from biology students in particular?
Biology students will think that I could do these experiments in the lab. I'm pretty sure, almost all lab in IIT Indore, all these experiments can be done easily. Okay.
Yes.
Okay. All, not a small thing. Only crystal structure you have to go to our CAT, which is much better indoor system we have. But basically, it's not about that equipment or something. It's about the logic way which you are looking for.
It is better to set a long-term and short-term goal so that you proceed in research in optimized way, a better outcome. In fact, I was thinking when you started talking about this sensory organs, which are very fundamental. In fact, as per physics, the universe is almost 13.8 million years old. And these things are so fundamental. These discoveries are coming just, recently. Initially, it was a bit surprising for me, but, looking into the internal dynamics, so essentially the technological advancement happened last 1-2 centuries. So that's no surprise, but I was thinking in that way.
Because the question is now coming to the biology or any high impact biology, that you have to show the mechanism. Okay? By mere telling that there is something is not enough, and you have to mechanism, somebody has to reproduce the data, and it takes time. For example, 2017 onwards, we see the cryo-electron microscopy is available. Okay? Earlier it was not even in India. If somebody has to do crystal structure, they have to try at least a couple of thousand log to get only one success. And nowadays that is vanishing out in India or elsewhere also. Okay, so everybody wants success in the past 2 attempts. So how it is possible? So when things are available now, we can do much more discovery.
And again, going back to my initial discussion, and addressing to our student community that look at the patients, thousands of genes, you have to clone that into independent vectors, that those many clonings,
Yes.
transcription into fibroblast cells, and then
Yeah.
asking that question that whether there is a gain of function.
Yeah.
So understand that 99% you have failure, but you are waiting for that one which is the right one.
Yes. Yeah.
You said that IIT Indore is the right place, and yes, I agree that these are simple experiments, and with these simple experiments and setup, one can really do marvelous discoveries here.
Yeah, I mean, point is that the technical nature of these experiments are well understood by all of us, biological student community also, and with little training of weeks, they can do the same experiment what when done. So basically, it is asking the right question, okay, and how long you can run in that race.
If there are no more questions, I would like to take this opportunity to thank Dr. Debashish. Thank you very much for enlightening us. You have explained this complex phenomena in very simple layman language. And I know these are very complex papers. I have tried to read some of them, and I thought that no, I cannot read and understand them in one go. But yes, in very simple language, you have explained this complex phenomena. So I express my heartfelt gratitude for accepting our invitation and spending your time to explain this particular thing. We would like to have you here in person soon.
Yeah, sure.
And then maybe we can have extended discussion on this and other topics. Dr. Raghu, if you want to add something, otherwise we can stop.
Yeah, in fact, if you see the whole idea behind this kind of outreach program, this has not happened overnight. In fact, we started planning because our next generation scientists and engineers are on board. So I should tell that, you see, the idea comes in the following way. As I told at the beginning in the welcome lecture, so research can be categorized as any path-breaking discoveries or fundamental research versus incremental research. And I also explained at the beginning that everything has its own importance in its own domain, because to understand the whole dynamics, incremental research is also sometimes becomes important. And people may be surprised why a particular work gets Nobel Prize, why not the other one. If you look into Nobel Prize winning works, in fact, Arnold Sommerfeld was nominated almost 84 times, and he could not get Nobel Prize, whereas his associates, 7 of his associates could get Nobel Prize. So essentially, recognizing the fundamentalness of the work is very important, and it is not always hour-centric. Rather, our brings forth this internal dynamics of this kind of works. And essentially from IIT Indore, what we wanted is this kind of outreach programs can initiate a different kind of interdisciplinary thoughts. And it was a tough job. Since we are ending the session, let me speak 2 minutes on this. It was a tough job of identifying eminent speakers all over India. And in fact, the planning started almost 2 months back, and today we have come to an end. And I should take this opportunity as the Associate Dean of International Affairs and being the convener of the outreach program of IIT Indore to thank our Board of Governors Chairman, Professor D.B. Phatak, Professor Nilesh Jain, our Officiating Director, and Professor Avinash Sonwane, our Dean, International Affairs and Outreach. So to be with us, which was very encouraging. In fact, everybody participated actively in this program. Although we wanted in offline way, so because of the corona situation, it was online, but online has its own advantage, like reaching out more people. So our organizing committee members, Professor Biswarup Phatak, Professor Ruchi Sharma, Professor Nirmala Menon, Professor Prasad Kodgire, so all are very active finding the eminent speakers, because as you understand, these fundamental discoveries has to be translated to more fundamental way that we can express and talk to general public across the disciplines. And it was a wonderful job by all the speakers, and we could get eminent speakers like Professor Rama Govindarajan from ICTS Bangalore, Professor Gautam Menon, who talked about physics discoveries, and he's from Mat Science, Chennai, and Ashoka University, Sonepat. And Professor Ravi Srivastav talked about economic sciences Nobel Prize. Then we chemistry, we covered chemistry Nobel Prize through Professor Devapalli Ramachari from University of Hyderabad. Then we had a session on Nobel Peace Prize with Nidhi Razdan, former NDTV anchor and director of Outreach and Strategy Program at Geetam University. And yesterday we had a good session with Professor Nishant Jaidi talking about literature. And I enjoyed a lot when the literature was associated with geography and history and how these things influence the writing of a person. And needless to mention that our ending is a wonderful ending with biology and connecting to physics and chemistry. So I enjoyed a lot. And at the end, in fact, the glory also goes to all our enthusiastic audience. And so you see, collectively we made this show as a big success. And I look forward to continuing this kind of program in the coming future from IIT Indore. And before we end, I can see our Dean International Affairs, Avinash Sonwan, is on board. If you want to speak a few words, then we finish the session. Avinash, if you are there, if you can speak few words as the closing remark.
As Raghu was telling, that we as a department and institute would like to have more such interactive sessions in future also, either online or offline, whatever is the feasible as per the circumstances. But understand that just doing science and reading whatever is there happening in your group is not sufficient to understand the breadth of the knowledge. Participation in these talks is going to make you a mature and seasoned researcher. So you will get ideas. You will also know whether you are going in right direction. You will know a lot of the technicals know-how. Therefore, I humbly request all the students, whenever you get an opportunity to listen to a talk, don't miss it. I think Avinash has some technical issue in connecting. Avinash, can you listen to us?
Yeah, maybe. Okay, so fine. So you see, collectively, in fact, I wrote to individual department heads. Since Prashant was active, I could not write yesterday, but I wrote to individual department heads to sensitize the students, because collectively only we can bring a vibrant academic atmosphere in any campus, and students are an integral part of any academic ecosystem. So looking into these things, I have tried to reach, which many people do not know, our student bodies, student Gymkhana, then our international affairs section of students, individual heads of the departments, that everybody should participate in this kind of public outreach talks. And personally, it is very important for me to note that, you see, as a physicist, we understand the different kind of physical interactions. And as I could see from Devashi's talk today, so both chemistry, physics, and biology, okay, so everything, all kind of fundamental knowledge one needs to have fundamental discoveries. So let's try together that, we come up with a vibrant atmosphere, academic atmosphere in the campus, which is slowly becoming important for any academic institute, not only IIT Indore. So yeah, so let's try together, because only Prasad, me, or Devashish, or anybody in the organizing committee cannot make it. It is the collective effort of the system. Okay. So I think Avinash cannot connect. Oh, he's there. He's listening to us maybe. Some technical problem from his side. So I think we have come to an end, and I guess I missed our international affairs section, particularly Riyanka, Suresh Thakur, Rajan Thomas, and Binnoy. So doing a wonderful job this time, taking care of all technical aspects. So I think they also need a special vote of thanks. So yeah, thank you so much. Yeah.
Thank you.
Thank you, everyone.
Thank
Thank
you,
you.
Devashish. Sometimes we look forward to see you.
Thank you so much.